Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, Developing Impurity Analytical Methods with a Quality by Design Approach. My name is Sarah Thomas, and I will be moderating today's presentation. I'm delighted to be joined today by Dr. Mark Argentine, Senior Research Advisor in the Analytical Research and Development Division of Lilly Research Laboratories at Eli Lilly and Company. Today, Mark will discuss risk-based approaches, including analytical quality by design and method lifecycle management that can be implemented to develop fit-for-purpose and robust methods that improve the control of impurities in drug products. After the presentation, we'll move on to the question and answer session. Please feel free to ask any questions during the presentation. You can do this by submitting them using the button to the left of your screen. Just click on the purple tab. So without further delay, I'd like to hand over to Mark and to thank him for presenting to us today. So please go ahead, Mark.
Well, thank you and welcome. Today I'd like to talk to you about developing impurity analytical methods with quality by design or an analytical method lifecycle approach. And in the process of doing so today, I'd like to briefly provide an overview again of an analytical procedure lifecycle, but more importantly, hopefully offer some tools for efficiency in analytical method development for impurities. Along the way, hopefully provide some meaningful considerations around establishing or maintaining a level of control within your method, particularly around defining a meaningful system suitability sample, and hopefully do all of these in construct of shared examples around these concepts for impurities methods that do leverage analytical knowledge in this life cycle approach, both in development and then ultimately with transfers and through method changes. Again, a lot of what I'm going to be talking about today is not new or revolutionary, but hopefully will provide you some useful tips going forward as you think about your needs with respect to impurities methods development and use. So an analytical method life cycle can be easily summarized in the three stages that have been outlined in this USP stimuli article that is cited below. And ultimately, a lot of work or time is spent in stage one associated with the design or development and understanding of what is needed to be measured. What analytes, at what levels, are all important aspects toward considering or appropriately designing an analytical method that's going to be intended as fit for purpose. And then obviously stages 2 and 3 are more around confirmation of performance and continued evaluation of method performance as fitness for purpose over time. Thought of another way or in a similar way is shown in this pictorial illustration, where we start with patient or product needs that leads toward development of an integrated control strategy. And that integrated control strategy from an analytical perspective can ultimately identify analytical method needs, and those can be developed. And there may be a period of time where we have development methods. That might be an early phase development for a new asset, or there might be a lot of information already for a reasonably established product, and that is some development within your laboratory for an existing product, toward defining what's necessary for ultimate control methodology. And then we move into stages 2 and 3 for qualification or evaluation of initial performance and continued improvement. Well, I speak about an integrated control strategy. One might say, well, what is that? Well, obviously, it is a combination of process and product development knowledge, and it really leverages both process controls and analytical controls to ensure the quality of the desired product material being made, whether it be a drug substance or a drug product formulation. And of course, this knowledge basis or integrated control strategy is really developed through a lot of thoughtful experimentation that happens over time. And some of this information may be available or it may be generated. For example, the use of design screens around force degradation studies will identify primary degradation mechanisms and primary degradation products that one might anticipate, as well as susceptible conditions for degradation, and whether they might be observed or likely given a product exposure. Other examples of experimentation can include multiple analytical methods, perhaps in a development mode environment that will leverage advanced technologies such as diode array mass spec, NMR, or other spectroscopic tools, multiple chromatographic methodologies that lead to understandings, particularly as we think about impurities. But most importantly, we think about this control strategy from an impurities perspective having both a knowledge-guided aspect and a chemistry-guided aspect. The knowledge-guided aspect would include evaluation to understand what impurities have already been observed in selected samples or materials. The chemistry-guided perspective might ask questions such as, what is possible or likely from an understanding of the synthetic process that may be utilized or the formulation process, the excipients and the potential impurities in those reagent materials or excipients that might be possible. And so a combination of both help to develop an overall robust impurity control strategy conversation. And then one can leverage this information to really help develop an overall understanding. And so an illustration of one of these types of tools is shown in this slide, where one might consider a broad gradient screening methodology that might span multiple synthetic steps in a drug substance manufacturer to follow formation and fade of potential impurities. And in this illustration, we have starting materials A and B going to an intermediate product AB converted to a subsequent intermediate, then ultimately reacting with a third starting material to form the final product. As we look through this particular example, we can see by evaluation of reaction samples some ability to monitor not only step 1 but also step 2, but we also then can look at isolation of materials such as the step 2 material and see that in general many impurities might be rejected, but the most significant impurities that can tend to linger include these A-related impurities or impurities associated with the A starting material. Nevertheless, most impurities are effectively removed through crystallization, and this may lead to some good, hearty understanding and assurance that we have good controls around process controls, for example, during the crystallization process, and analytical can be simply a verification assessment. This information can kind of be captured in other ways as we perform fade and purge studies through experiments just like I had mentioned, to then look at different understanding of impurities with an end in mind. In this particular illustration, I'll highlight, or I've highlighted 4 impurity scenarios. I'll only mention a couple here. For example, impurity C might be an impurity that showed up in earlier toxicology material and has been qualified at a level of not more than 0.25%. That impurity actually is introduced in the penultimate step, and we know that there's about an 80% removal potential during downstream purification or reactivity to the final step. As such, one might consider a control such as not more than 0.5% at the step 2 stage to then ensure that we have suitable control in a conservative format and ensure that our impurity C remains below the not more than 0.25% limit in the final product material. In an alternate case, there may be situations such as impurity A, where impurity A is perhaps maybe not typically detected, but it is potentially present or possible, could have been observed through, in this particular case, maybe a reaction or a mother liquor sample to show that it was present, but present at low levels, and we know that the rejection efficiency is so high, greater than 98%, that it is very easy to routinely control that impurity well below a specified impurity level, and as such may not have any special controls even at the step 2 intermediate. So again, part of these designed knowledge-based studies that happen throughout development on a specific process or condition help understand key questions. What impurities, at what levels, and what precision is needed to design an appropriate analytical method? Well, all those questions really speak toward an analytical target profile for what is required for a fit-for-purpose analytical method. And that can help frame our conversations going forward. Again, to probably reiterate a point that I've almost already made, we can leverage a broad variety of development tools and methods to gain this process knowledge. And ultimately, some of those tools may be more sophisticated. And as we think about long-term controls, we also then think about a transition to the long-term control strategy associated with an asset. And that will be integral to its specifications or controls, and of course is also optimized for ruggedness and performance in a routine control type of environment. And so as one starts to develop or build an impurities control methodology, one can then categorize impurities. In this instance, or an example I've done for drug substance impurities control, one can think about breaking these impurities into different tiers of consideration. Tier 1 might be the must-have impurities. Those would be those from your designed experiments or from your practical experiences have been observed impurities or very likely impurities that can have potential to propagate, into the final drug substance. They would also include primary degradation products for a stability indicating methodology. And certainly I've shared some examples of those types of impurities, whether they be formed or observed in reaction samples in mother liquors. A second tier would be those nice-to-have impurities, and those would be impurities that one would consider or be nice to have in a single analytical impurity method for control, but perhaps there are alternate analytical controls already in place or as an option. For example, one might have regioisomeric impurities in the drug substance from potential regioisomers in the starting materials. But if one already has an analytical method developed to control for potential regioisomers in the starting materials, then control in the drug substance material may be less critical or already managed through upstream controls. And then lastly, tier 3, those impurities perhaps not needed or excluded, might be those impurities that were only potential impurities from a chemistry perspective and never found to be possible, or impurities that are separately controlled, such as an enantiomer of a small molecule API, where there might be a separate chiral test method for that type of control. With that type of impurity collection or understanding in place, one might then say, so where do I start? Well, one can certainly use systematic tools and models to aid in appropriate method development. And what's shown here is if you happen to know those potential impurities, you can use models to plot pH versus log D to also provide some direction. In this particular plot, I've captured both acidic and basic components, and you can see that more often than not, we apply low and or high pH spaces as a good starting point to deliver both some separation performance, but also some robustness around chromatographic consistency. We can also leverage some very useful development tools, and that includes automation as well, particularly in column screening. There are several tools that, for example, utilize diode array and mass spectrometry with some automated peak tracking using both UV and mass spectral signals to help guide very efficient methods development. So ACD Laboratories, for example, has a suite that is very helpful for doing so. And as well, the Fusion-based software has recently added this mass spectral capability in addition to UV tracking for efficient tracking performance. Other tools that may be leveraged include some items such as advanced software and automation capabilities that may be used for not only development screens, but also for robustness studies. There are several different software opportunities for such. I've highlighted just one with the Fusion software, but there are method development or method validation software tools that can be helpful for designing appropriate experiments in a quality by design type of fashion, automatically building sequences and instrument methods. In this particular case with Fusion, very readily with the Empower-based chromatographic data system, where systems or designed experiments can be initially established, the samples or series of samples executed, and once data are processed, data can be automatically retrieved back through the software system for automated analysis reports. Those reports are very helpful and provide great visuals for understanding from a model-based perspective, perhaps some robust spaces for routine method execution. In a similar format, other approaches can involve the use of deliberate column screening with columns that offer different separation mechanisms and separations at both low and or high pH, perhaps even with different solvent systems, to look at different distinctly separation conditions. From those screens, one can look at items such as selectivity or purity peak spacing, as well as peak shape, to pick good leading opportunities for further pursuit. And one can then leverage additional separation, particularly with tools like modeling, especially for small molecule, leveraging tools like DryLab or LC Simulator, or with empirical models such as through the Fusion software when working with multi-point modes of interaction, to really guide additional separation development and predictions for additional directional studies. I kind of take a quick aside to mention the reminder that not only does one get some good information from these modeling tools around desired or predicted conditions for best operation, but one can really also understand some robustness around, or likely robustness around, an analytical method with these tools. So in this particular heat map that does capture parameters such as temperature, organic strength, and gradient sensitivity, one can see in this particular case if there was a resolution or a minimum resolution that needed to be at least 1.5, then really any place in green all the way to red would be favorable. And so there may be, in this particular case, good examples where there's a broad region that is acceptable both in temperature and gradient sensitivity for developing robust condition. And a reminder that these models are very good at predicting some actual separations if they're appropriately applied. Importantly, as we think about then not only defining the chromatographic conditions, we want to think about additional controls and important attributes around the actual analytical procedure itself. And obviously, some attributes that are important for an impurity analytical method would include specificity, detection, precision, and accuracy. And we can see that several of these attributes can be at least probed even on a regular basis through meaningful system suitability attributes or aspects. And I'll talk a little bit more about that in the future. But certainly a reminder of the importance of system suitability capabilities for monitoring performance as well and helping to ensure robust analytical methods. As we think about moving toward the latter stages of development and the life cycle with procedure qualification, I would like to suggest that robustness and system suitability, while listed in this table here, are really best designed and performed prior to procedure performance qualification, really to ensure robust method performance with meaningful system suitability incorporated within those designed experiments to ensure that one has a robust method as well as robust system suitability controls in the final analytical procedure. So let's move to a couple of examples that hopefully illustrate some of the points that I've just mentioned. In example 1, I have a trace impurities limit test where a drug substance is isolated as a tosylate salt. And in this particular instance, there were 2 alcoholic solvents that were present during that final API isolation processing step, and they could form tosylate esters. Of course, those tosylate esters are potential mutagenic or genotoxic impurity, and they would require appropriate low-level control, in this particular case at not more than 2.5 ppm each in the isolated API. Well, the method was initially developed in this particular case with dry lab assistance for these small molecules and that matrix to support a robust method. And initially there was some work to demonstrate in early development some good linearity from 1 to 50 parts per million for each impurity, trying to understand potential levels but also rejection potential if present. But ultimately, the desire was really a limit test as a routine impurity control, since we didn't expect the presence of any of these impurities on a routine basis. And that can streamline overall simplicity and robustness for intended use. So what might that look like from an ATP, analytical target profile? One would want to confirm that the impurities X and Y are below 2.5 parts per million each in the isolated drug substance material. In this particular instance, you can see some of the requirements around an analytical methodology and performance. But most importantly, the analytical method capability in this instance can be confirmed each time the method is performed with an appropriately defined system suitability sample. One can utilize a reference standard, in this case a reference standard that doesn't contain these 2 impurities, spiked with 2.5 parts per million of each impurity, so an impurity at the limit of concern. And that can ensure acceptability of the chromatography from a selectivity as well as detectability of each impurity in the matrix prior to the evaluation of each test sample. And that example chromatographic overlay is illustrated here, just showing an overlay between the blank 2.5 part per million standards of each, and then the 2.5 part per million standard of each spike in the impurity matrix, an acceptable recovery from the matrix itself. And in this particular case, a simple limit test, in fact, even with a UV-related detection methodology, was put in place for routine types of control. In a second example, we might again be interested in trace level impurities, but now in this case, more interested in a quantitative control. In the second situation, there were actually two potentially mutagenic impurities present in late-stage intermediates. And there was some development data collected to show that these impurities had purge factors of at least 80 for each impurity to the final API. Well, that required an implied control of about 600 parts per million at each intermediate, because I believe that the overall control was at 7.5 parts per million in the final API. But the desired level of control might be at something a little bit lower, perhaps at 500 part per million of each impurity at the intermediate, to provide a conservative control at the intermediate stage to control these impurities. But also maybe the ability to monitor for impurity formation at lower levels, maybe down to 100 parts per million, based upon, in this particular case, there was some process experience that would suggest these impurities are not likely to be present at significant levels through upstream removal potential. And as such, want to potentially develop a case for information and potentially even greater control by demonstrating the absence of these impurities at, say, 100 parts per million in the intermediate. So a method was developed with modeling assistance to support a robust method with 100 part per million quantification. An analytical test target profile might look like this to control compounds X and Y, again, two different impurities, but below 500 parts per million with a tolerance of 20% in the isolated intermediate material, with the capability to quantitate to a level of 100 parts per million, or 20% of that 500 ppm limit. And we can understand some typical acceptance criteria to ensure adequate performance for that type of expectation. Indeed, an analytical procedure was again developed. You can see a chromatographic overlay here, which shows an opportunity where the matrix was removed fairly quickly under some chromatographic conditions and allowed these two alternate impurities to be separated well, and that even at the impurities at their target limits have very good detection sensitivity. As we may see from qualification-related results, we could readily achieve outstanding signal-to-noise capabilities, even at the lowest of levels, with good precision and accuracy or recovery of those impurity standards on a routine basis. As such, the method was certainly appropriate, and one can define a meaningful system suitability to ensure acceptable routine performance. In this particular case, obviously the blank can be routinely inspected to be free from interference, but authentic standards are available for these impurities and could be routinely prepared at the reporting limit, and of course shown to have adequate signal-to-noise and acceptable resolution between the two impurities to accurately separate and quantify those components. And then, of course, precision on those standards can be put in place for reliable instrument performance as well in the quantitative work. And finally, in a third example that's probably more typical of a more general organic impurities methodology with quantitative control, we have a drug substance that was crystallized in high purity, but there were several potential process-related impurities when looking at, for example, mother liquor samples. So the crystallization process was very fortuitous and giving high purity material. But one wants to be able to protect against potential issues with crystallization. Additionally, the drug substance was stable, but there was a hydrolysis potential at pH extremes. To add further challenge to this particular development, there really were no qualified impurities since the drug substance was always crystallized in very high purity. That then suggested that there was going to need to be routine control of individual impurities below a specification limit that was one for any individual specified impurities. since there really were no qualified impurities above the individual specified limit. And as noted here, there were several, at least 10 process batches that supported the fact that control with all individual impurities at perhaps not more than 0.10% for each impurity was possible. So again, a method was developed with modeling assistance, as I've highlighted with previous examples, to support a robust method with quantification down to the 0.05% reporting limit. One can generate an appropriate analytical target profile for such a type of method. In this particular case, again, the anticipation would be for no specified impurities or control of each impurity at the ICH unspecified impurity limit. And so work was done with designed experiments. And in fact, this slide illustrates a dry lab optimization video that's running not only for what was generated through a design study for separation of these components, but again, a reminder of the robustness in information that can be gathered associated with this particular separation. In this particular case, of course, yellow or higher in colors are better, and one probably doesn't want to run a method that has a very, very long gradient time that might be seen on the far lower right, but have a more efficient, from a time efficiency perspective, analytical method. Well, from this, we can look at our modeling-based data to see where our critical pairs are. And in this particular case, there's a critical pair that is noted or circled, and we can see that it has sensitivity with respect to organic content and gradient time, but is not very sensitive to temperature. And so we can understand that particular need. We can understand what those impurities are. This became particularly important in this situation because those impurities happen to have response factors that are actually a bit different than the API itself. And so we wanted to particularly understand or make sure that we have resolution and tracking for those components. Well, we could have looked at our optimized dry lab conditions parked in the middle of that yellow zone and found that we had gotten very good separation. And in fact, this slide illustrates that we were in fact able to get good separation for components. The critical peak pair is listed here that simply highlights that separation. Well, what's also interesting to note, perhaps, is that there does also appear to be another impurity that happens to be at the rear tail of the main peak. And so one could think of that as potentially a critical pair as well. The modeling data suggested that that peak and that separation is very robust and not sensitive to temperature or gradient conditions within the region of method development that we're looking at. And so that is probably not as critical a pair as the first, but wouldn't it be nice to have some information to support that position? And so a system suitability mixture of the two critical peak pairs were defined prior to robustness studies. Again, a well thought out, designed set of experiments for robustness can be executed even in a semi-automated type of format or fashion. You can see the type of parameters that were studied in this particular attribute that did include aspects of organic content and flow rate pieces that have impact to grading conditions. And from that, we can then evaluate our system suitability robustness results. And what we find is, as expected from our modeling information, that critical peak pair 1 is in fact fairly sensitive. The lowest resolution happening under a set of experiments that did have very high gradient-related organic conditions that we would have predicted would have minimized that resolution, and we saw that drop as low as 1.1. But in all other instances, resolutions were 1.5 or greater, and in fact, as typically as high as 2.5 to over 4. Meanwhile, the peak pair 2 really was fairly high to begin with and really didn't change throughout those entire set of experiments. So in the end, peak pair 1 was understood as most critical from our modeling and re-verified through this particular assessment. And really then, from that knowledge, a meaningful system suitability could be defined. First and foremost, the suitability with a minimum resolution of not less than 1.5 for critical peak pair 1 could be defined. We know that sensitivity to organic content or the gradient is important. And so if there were any failure in the resolution, instructions around evaluation of either pump mixing or mobile phase preparation would be likely root causes to evaluate in the case of a system suitability failure. But it's likely that system suitability should not have any issues with failure under that minimum resolution of not less than 1.5 based upon the robustness study design. Additionally, we can add those key impurities as part of the routine system suitability mix so that we continually monitor that resolution, and we have the added benefit of helping to aid with positive peak identification for each of those 2 impurities. And again, that's important when response factor corrections are necessary and to ensure that we have proper peak assignments when response correction fractions must be assigned. So again, all these can be incorporated as additional method controls, and then we would apply the other standing detection, sensitivity, and injection precision controls for routine control and performance. Since I mentioned a comment around response factor correction, thought I'd take a moment just to briefly and continue to remind folks that looking at spectral profiles around impurities is important to make sure that as one's defining the desired detection wavelength, one considers those factors. But there may be instances where wavelength robustness may be an issue based upon even knowledge of that spectral information. And this example that is shown here, and if you want more information, you can see the J-Chrome-A article in which this is based, but impurities that may be even structurally fairly similar to the API may have very different spectral responses. And in this illustration, a 1 nanometer difference in wavelength has a fairly significant impact on results for individual impurities. And a 1 nanometer wavelength is typically well within or tighter than some of the typical instrument wavelength accuracy qualifications that are routinely performed. And indeed, you can see the reasons for those sensitivities. And if one normally picked, as one might like to, a wavelength for the method that is at the maximum of the API, well, that would be way out at about 305 to 310 nanometers, which would be fine for the API, but one would have essentially no detection sensitivity for a couple of the impurities, and they would be missed. On the other hand, you could pick lower wavelengths, but then we might not have sufficient sensitivity for impurities that are API-like in nature. And so a compromise had to be made, but of course, in this particular case, at 280 nanometers, there are some spectral responses that have profiles sloping in opposite directions, and hence the very significant difference in response factors if one's looking at the API as a reference relative to other impurities. And so one solution could be a simple wavelength system suitability with commercially available materials to ensure wavelength accuracy and consistency in your analytical results or reporting. There are other strategies that can be applied with individual authentic impurity standards for quantification as well. Again, just a reminder that one needs to continually look at wavelength spectral profiles and wavelength robustness as well when considering analytical methods. As additional reminders, not only once one's developed analytical methods, don't forget the capabilities of routinely looking at method performance data to continue to monitor analytical method performance over time. So as methods are defined, one might be able to readily capture with chromatographic software tools today, like Empower and many others. We have easy access and capability to extract information such as system suitability attributes from method performance samples or results to really monitor overall performance and variability of an analytical method. And it's just kind of a reminder that method performance sample data, if you've collected this information while that method is being established and qualified, that can provide some very useful information or a data set to support method transfer as well. In this particular instance, I just highlight an example where tracking, in this particular case, peak tailing that was monitored as a routine system suitability check on a method that historically was running on the order of about a peak tailing of 1.2, all of a sudden then ended up running higher to 1.3 to 1.4, and a limit of 1.5 was established for the method based upon earlier development. Well, upon investigation, one could look, and that turned out to be a movement to analyses on a couple of new pieces of equipment or instrumentation, and the instrumentation hadn't been appropriately passivated, and that affected the actual tailing and performance. So again, simple tools such as tracking performance data can rapidly help in addressing observations in analytical method performance as well. In terms of method transfer, obviously from an impurities perspective, there are certain considerations, but overall impurity method transfers should have an assessment, obviously, for the capability of that receiving laboratory to perform the analysis. But not only is it that assessment of performance of the method, but also one should remember that there's a lot of knowledge that and assumptions that are built behind that particular control strategy. And the transfer of the process and product knowledge in that overall control strategy is important context for the intended use of that method in another laboratory. And so, again, this also becomes very important for that receiving laboratory in terms of its understanding of proper method use and, as well, background information as reference for any future potential improvements that may occur in the process and implications that may have on the analytical method. In terms of some tools for method transfer, again, one can speak to toward data that I've mentioned, any data developed from robustness studies or method performance samples that have been captured in earlier studies prior to transfer, as well as any other experiments that have been collected through development to understanding risk and change. Additionally, through method transfer, there's an important desire to have confidence in the method performance, and ideally from an impurities perspective, method performance across the range. A lot of times this might imply the use of impurity-rich samples for meaningful assessments, which sometimes may be difficult for, as I mentioned in one example, high-purity drug substance samples. How does one appropriately ensure transfer and capability of the method to perform adequately? Certainly system suitability samples would be a good tool for ensuring appropriate method performance. Alternate or additional options include the use of perhaps impurity-rich development materials or samples that were deliberately stressed to provide impurity-rich samples, or perhaps stable product materials even spiked with impurities. All of those can help provide even greater confidence in method performance not only on typical materials, but also on materials that may show some sign of impurity formation and or degradation. This may be even important for less stable materials. For example, in larger molecules, such as peptides or more, one may choose strategically to use stress stability samples or a stressed material that might be representative of degradation at the end of a shelf life for transfer, along with meaningful transfer acceptance criteria based upon method performance in the development laboratory. The next slide just kind of illustrates that as perhaps an example where a material at batch release might have a very low impurity level load, but one could challenge that through a transfer sample with a higher level impurities, such as one observed after, say, 6 months of accelerated stress condition that might be at least representative or more worst case from that at the end of a shelf life. There's a lot of value in using a compound that has some degradation throughout the shelf life, simply because the method can continue to demonstrate that performance of the methodology is appropriate in the laboratory, not only to support release, but also throughout stability. And it's an opportunity for the receiving laboratory to analyze a more degraded sample that they may have never seen, and an opportunity to discuss some of this knowledge and understanding of anticipated or likely impurities, their formation and controls as part of that transfer discussion. And then last, as we think about life cycle opportunities, we can think about opportunities or cases where there may be potential manufacturing changes or method changes. In this particular case, we talk about method changes. If studies have been defined to understand that knowledge base well, and they're captured, one can leverage that historical knowledge and historic methods to use and evaluate for continuous improvement opportunities, and really either reconfirm the suitability of existing methods and controls for their intended purpose, or perhaps recognize that there are potential concerns. And I'll share with you an example here in a moment of the use of a gradient HPLC during earlier development studies, as I highlighted earlier in early development, broad-based to gradient methodologies that can be utilized in one particular case versus use of an isocratic method that was ensuring appropriate evaluation for impurities from an initial route of synthesis and developed to be a very robust methodology, but may or may not be appropriate as process changes occurred. So that example is really there was a method change that was desired in that particular case. After there was a manufacturing process change to also detect a potential new impurity, I'll call it impurity 7, where this gradient developed method that was available in early development was able to see impurity 7, but the routine isocratic impurity method was not, because impurity 7 was not relevant in the defined process chemistry prior to the process change. But as a result, there was a need to change the assay and impurity methods, at least the impurity method, that perhaps were more appropriate and capable for the new manufacturing process. This happens to be just an illustration of that isocratic impurity method 1 that was in place prior, and simply shows that process and degradation-related potential impurities were well resolved in the isocratic impurity method initially established, but impurity 7, which became a new process impurity, would not elute during the initial isocratic conditions, and obviously that was modified to a newer set of chromatographic conditions. That newer set of chromatographic conditions was developed to not only separate the existing impurities from the previous route, but also to include impurity 7 in, again, a newer set of isocratic robust conditions. And that now new method can see all the impurities of the previous method and process as well as the new potential impurities. So this allowed kind of consistency and a single new method to be utilized for evaluating and ensuring control of the historic method material or historic manufactured material as well as the newly manufactured material as well. And again, comparability assessments could be made to show equal control and performance, including evaluation of an impurity-rich sample by both methods, since in most cases the impurities were not typically present. So again, one can look at not only total impurities, individual impurities, and largest individual impurities in detail to ensure confidence and comfort in making a transition. In other instances, perhaps one simply wants to leverage new technologies for improved lab efficiency without necessarily a process change. In this particular illustration or instance, there's a condition where the separation was initially developed on a 3.5 micron particle size, 3.5 micron, 150 millimeter diameter chromatographic column. And you can see that the assay worked perfectly well, provided good separation and control for impurities. Obviously, one had level or control, and from a typical perspective, impurities that were well resolved from the main peak. But there was a desire to move toward a sub-2 micron column, shorter column for overall laboratory efficiency. And while there were, while this gradient and the injection volume were scaled appropriately, one can see that while there were some slight changes or slight decreases in overall resolutions that were achieved between the initial method and the newly proposed method, certainly the resolutions and capabilities were more than adequate for providing routine control for impurities with the new analytical method set of conditions. So again, with the mindset of that in mind, one can perform comparability assessments and proceed. As such, as we think around an analytical life cycle, hopefully what I've kind of shown you is that through earlier experiences, one develops this broad knowledge space. There may be an investigational method or series of methods exploring for potential impurities from varied sources and conditions that then can slowly get streamlined or simplified toward actual or likely impurities that are observed. They can include primary degradation products and may constitute what would likely be a more focused design space for routine control. And those can be optimized for routine impurity method control and may even at some point be optimized even further for design for speed, robustness, or other considerations. With a reminder that in all cases, the focus for these development methods and controls are fitness for purpose, and there are assumptions that these methods that have been developed are developed with specific process and control points in mind based upon that process knowledge. So hopefully, through this presentation, I've highlighted or illustrated that the analytical procedure or life cycle approach that is very methodical and integrated gives credence toward a good requirements-based approach toward method development and implementation, where analytical method development happens really in two parts. The first, a collection of knowledge, of overall process understanding and defining those key integrated process and controls, both analytical and process related. Those help to define those fit-for-purpose analytical methods. And effective method definition with meaningful controls like system suitability put in place help to continually evaluate and ensure proper method performance, ensuring that one is monitoring for those attributes on a consistent basis as well. A reminder that method robustness and modeling can greatly help with meaningful controls and with continued method lifecycle assessments. And lastly, a reminder that, again, from an industrial perspective, as you're developing that control strategy, you can see that from the conversations I've made, those integrated control strategies are specific to a process or a formulation that's being commercialized or controlled, and that those analytical methods are tailored for that combination of process and analytical controls for that specific process. And of course, any changes to process require reassessment for continued suitability of the analytical method or changes to the methods as they dictate changes to the product control strategy. So again, hopefully this provides some illustrative examples that are useful for evaluating and monitoring impurities in your analytical methodologies over a method life cycle. With that said, I'd like to certainly acknowledge some contributions of some colleagues on this presentation, and I'd like to thank you for your attention, and certainly would be welcome to entertain any questions that you may have.
Thank you very much, Mark, for your informative presentation. It's now time for the question and answer session. We'll try and get through as many questions as we can. So the first question here, the case studies that were shared appeared to provide focus on chromatographic aspects of small molecule drug substances. Can you comment on the applicability to drug products? Are there any additional considerations that you might be able to share?
Sure, and thanks for the question. Yeah, certainly, hopefully I've shared that these strategies are readily applicable to drug product methodologies, particularly small molecule ones, in fact, with perhaps a comment that methods usually will focus or change to focus more on drug products, as I've highlighted, and or formulation-related components or impurities that are more important in a drug product impurity control strategy. Probably also important to stress the importance of sample preparation in drug product methodologies and the recommendation for thorough robustness studies regarding sample preparation as one thinks about drug product in addition to the chromatographic robustness aspects as mentioned.
Great, thank you, Mark. The next question we have here, the tools that have been shared, for example, chromatographic modeling and design of experiment software for development, aren't necessarily new. Where do you see opportunities for advances in these tools?
That's again an excellent question, and I agree. These advanced tools such as ACD Labs and more recently S-Matrix and others with models, but particularly ACD and S-Matrix offer some opportunities for peak tracking, and automated peak tracking is tremendously valuable. Currently, most of that software, particularly from an automated perspective, is limited to single quad mass spec, whether that's a QDA or a single quad itself, in the integration and automation. Clearly, there's opportunity to expand that support for more automated capabilities, particularly in the area of peptide and protein development. And while example, ACD Labs can import some high-resolution mass spec data, it currently can't control or manage those systems in an automated format. And there's also been enhancements in the user interface and reporting that have come a long way. But I think as we continue to look toward the future and evolution of varied modalities and studies, and obviously with peptides, the opportunity to greatly reduce time spent with development through verification on peak tracking can't be understated, and I think efforts in that area will certainly be encouraged and found, as well as leveraging these enhanced chromatographic prediction models to really account for things like peptide protein sensitivities and folding, which I know some modeling programs have and will only continue to become more important, as well as empirical models for that type of work.
Great, thank you, Mark. The next question. In your last couple of slides, you indicated the ability to leverage advanced chromatographic technologies for increased lab efficiency. It appears that one of the greatest challenges or barriers to this appears to be managing global regulatory changes. What are ways to facilitate enhanced regulatory flexibility and the method changes for enhanced lab efficiency?
Another great question. And indeed, scientific kind of strategies or method comparisons for moving from one to the next oftentimes make good scientific sense and can be adequately supported. However, regulatory management of those changes might not always be. And certainly, I think that's been one of the conversations and goals regarding analytical target profiles in this advanced method lifecycle topic. It's certainly hopefully an opportunity with the evolution of ICH Q14, which is currently being developed, perhaps in step one, and hopefully we'll see more of that in the future. But I think there's opportunity to leverage those concepts to support multiple acceptable methodologies that might be capable for an intended purpose. One maybe good example of kind of where that might be seen is in a recent example in Farm Europa 32 on nitrosamine detection in sartans as a limit test. And there were three different methods, an LC-MS-MS method, a GC-MS method, and a GC-MS-MS method. All three appropriately validated for one or several of the several common small nitrosamines to in the varied sartan matrices to a 30 part per billion limit. And so again, I think it provides an opportunity to say that there's not just one right analytical methodology to meet an intended need. And as we continue to work through these concepts to help with regulatory flexibility, that will be beneficial.
Great. So we've had a few questions here about will we have access to the PowerPoint presentation?
Yes, and I believe that this presentation, and I apologize for some if you had difficulties with connection today or not, hopefully not, but I know in this current environment with a lot of virtual meetings, some connectivity may have been hampered, in which case I think that this presentation will be made available on the Select Sciences website or otherwise, and so for registered attendees, we'll make sure that we have or communicate a way for you to be able to access this presentation after this live presentation.
Great, yes, I will share that on the Select Science website. Another question here, ICH Q14, Analytical Procedure Development and Revision of Q2R1 Analytical Validation dated 14 November 2018. Is there any news about this document?
I suppose the only update I can share is that the ICH expert working group has begun to discuss this topic. They met in Singapore back in November, and I think they were originally planning to have and prepare a step 2 document at their meeting in May of this year. I believe that meeting has been postponed due to our current global circumstances, but I do believe they're working as quickly and efficiently as they can toward getting a document out for step 2 comment later this year, if possible.
Okay, thank you, Mark. We've got time for one more question. How can you decide to use a limit test instead of a quantitation procedure?
Well, that will really depend upon what the need for your control strategy might be. Maybe to summarize, or in some instances, the examples I shared today may be limit tests, particularly when you're trying to demonstrate the absence of a particular component at a low level. So I've mentioned some examples with mutagenic impurities, and one might find that as an effective strategy. But there may be other good reasons when a limit test to simply demonstrate that a component is below a certain level is adequate for its intended purpose, in which case a limit test may be appropriate versus a quantitative test, which might be more helpful, particularly when tracking and trending results from batch to batch over periods of time.
That's all we have time for today. So thank you very much, Mark, for today's informative discussion and presentation. And thank you to everyone joining us online and for sending in your interesting questions. If you do have any other questions, then please feel free to email me at editor@selectscience.net and I will follow up with your questions for Mark. And if we didn't have time to answer your question today, we'll follow up with that after, so you have your answers. So you can also download a certificate of attendance in the related resources tab to the left of your screen. And if you'd like to listen to today's webinar again or to invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye and thank you to everyone joining us.